Morning students, in last session we discussed about the most important cell organelle, that is flagella, and we also studied the motility. Now today we have to discuss the another important cell organelle, that is Fimbriae and Pili. Okay, this Fimbriae and Pili, they are present on the surface of the cell as a fine hair-like appendages. Okay, many prokaryotic organisms have short, fine hair-like appendages that are thinner than the flagella. We already studied one of the cell organelle, that is the flagella. That is also hair-like appendages, but these hair-like appendages, they are bigger than the Fimbriae and Pili. Okay, so these appendages that are thinner than the flagella. When we see the width of the flagella, that is more as compared to Pili, and that's why Pili, they are thinner than the flagella. Okay, these are usually called as a Fimbriae, or singular, that is called as a Fimbria. Fimbriae and Pili are filamentous structures composed of protein that extend from the surface of a cell and can have many functions. A cell may be covered with up to 1000 Fimbriae, but they are only visible in a electron microscope due to their small size. These Fimbriae and Pili, they are very smaller in size, and these Fimbriae, they are visible under electron microscope only due to their small size. Is it okay? Now see, here is the photograph of the bacterial cell, and on that bacterial cell there is a small hair-like appendages. Okay, and these small hair-like appendages, they present on the all over the surface of bacteria, and they are present in higher number as compared to flagella. Okay, and these Fimbria, they are smaller than the flagella. Is it okay? That means it is clearly visible in this photograph. Here see, this is the flagella, which is bigger in size. That means the width of the flagella, that is bigger, and length also more as compared to Fimbriae. Okay, and now these are the Fimbriae. That means these are the filamentous structures, and they are present all over the surface of the bacterial cell. Okay, now they are cylinder tubes composed of helically arranged protein subunits and are about 3 to 10 nanometer in diameter and up to several micrometers long. Okay, means here the diameter is 3 to 10 nanometer in diameter. Okay, width is 3 to 10 nanometer. Okay, and smaller than the flagella. At least some types of Fimbriae attach bacteria to solid surface, such as rocks in stream and host tissue. Host tissues. See here, these main function of the Fimbriae to attach that bacterial cell to the solid surface, such as rocks, and sometimes in streams and on host tissue. That means for the attachment of that bacteria to the host body or any solid surface, these Fimbriae, they helps. Is it okay? Now Fimbriae, they are responsible for more than attachment and 4 Fimbriae are present at one or both poles of the bacterial cell. They can be aid in attachment to object and also are required for the twitching motility that occurs in some bacteria such as Pseudomonas aeruginosa, Neisseria gonorrhoea, and some strains of E. coli that bears such a type of Fimbriae, and these Fimbriae that will help to move that organism or to show the twitching type of motility. Is it okay? That means generally the organ which is responsible for the movement from one place to another place is actually the locomotory organ, that is the flagella. But in some organism, these Fimbriae also helps for the twitching motility or gliding motility. Okay, and specifically such a type of motility that is observed in Pseudomonas, Neisseria, and Escherichia coli. Is it okay? So not only the attachment but also for the twitching movement, these Fimbriae that will help. Okay, movement is by short intermittent jerky motions of up to several micrometer in length and normally is seen on very moist surfaces. That means such a type of twitching motility that is observed on the surfaces which are moist. That means that will help to move or twitching movement in case of only moist surfaces, and there is evidences that the Fimbriae actively retract to move these bacteria. That means in addition to the flagella, there is a Fimbriae that will also help to show the twitching movement of bacteria on some moist surfaces. Type 4 Fimbriae also involved in gliding motility by myxobacteria. Means with this they also helps to organism to show the gliding motility. These bacteria are also of interest because they have complex life cycle that include the formation of a fruiting bodies. These bacteria are also of interest because they have a complex life cycle and that include the formation of fruiting bodies. We know that there are some fruiting bodies or nodules which are formed in the leguminous plants. Such a type of fruiting bodies they are also formed in the such a bacteria. Now another organ which is present on the cell surface of bacteria, that is the sex pili, and many bacteria have about 1 to 10 sex pili, and singular it is called as a pilus. That means near about 1 to 10 sex pili they are present per cell. Okay, and they are helpful for the transfer of gene or plasmid from one cell to another cell. That means it is one of the mechanism through which there is a gene transfer that will be takes place, and these are also hair-like structures that differ from Fimbriae in the following ways. That means some microbiologists they consider the Fimbriae and pili they are same. Okay, only words they are changed, but it is not true. Here pili and Fimbriae they are different. Okay, so pili often are larger than the Fimbriae. These pili often larger than the Fimbriae. See here, flagella they are larger. Then below that there is a pili, then Fimbriae. In Fimbriae they are smallest structures. means whatever the hair-like appendages which are present on the bacterial cell, these three appendages, that is the flagella, pili and Fimbriae. Okay, and these Fimbriae, they are smallest than the two other thread-like appendages. Okay, now pili often are larger than the Fimbriae and it has around 9 to 10 nanometer in diameter. Is it okay? We already seen that these Fimbriae, they are 3 to 10 nanometer in diameter, but here pili, they are 9 to 10 nanometer in diameter in width. Okay, they are genetically determined by conjugative plasmids and are required for conjugation. In gene transfer mechanism, there are three different types of mechanism. There is a transformation, conjugation and transduction. And among that, the conjugation, that is one of the most important gene transfer mechanism, and in that here pili, they plays an important role to form a bridge between donor and a recipient cell, and through that bridge there is a transfer of these conjugative plasmids from one bacteria to another or from donor to recipient cell. Is it okay? Now some bacterial viruses attaches specifically to receptors on the sex pili at the start of their reproductive cycle. Viruses, we know that these are the parasites. Viruses, they are parasite and they are totally dependent on their host. The viruses which have an ability to infect bacteria, such a type of viruses, they are called as a bacteriophages. And some bacterial viruses, they are attached specifically to the receptors on sex pili at the start of their reproductive cycle. That means the first step in the virus reproduction is the attachment, and that's first step of attachment that is takes place on the sex pili. Okay, now the another one of the most important cell organelle, that is the nucleide or nuclear material which is required for the cell. That means each and every living cell that bears some nuclear material, and in case of bacteria there is no well developed nucleus, and that's why that nuclear material, that is called as a nucleide. The nuclear material of a bacterium comprises of its genetic material or nucleic acid, that is the DNA in the nucleoid. The nucleoid meaning nucleus-like structure. It is not a perfect nucleus, but it is nucleus-like structure which is present in the bacterial cytoplasm, and that is called as the nucleoid. This nucleoid is an irregularly shaped region within the cell of the prokaryotes that contains all or most of the genetic material. That means DNA. Okay. In contrast to the nucleus of eukaryotic cell, it is not surrounded by nuclear membrane. Now what is the difference between nuclear material which is present in prokaryotic cell and in eukaryotic cell? So in case of eukaryotic cell, the nuclear material that is surrounded by nuclear membrane, but such type of nuclear membrane that is absent in the bacteria or in case of prokaryotic organism. Okay, the genome of prokaryotic organisms generally is a circular loop double-stranded piece of DNA, of which multiple copies may exist at any time. This is the circular loop. Generally this DNA which is present in the bacterial cell, it is the circular loop And we know that the DNA, that is the double stranded. So in case of bacteria also there is a double stranded piece of DNA of which multiple copies may exist at any time. That means it is having self-replicating power. So the length of genome varies widely but is genetically at least a few million base pairs. That means based upon the size of the bacteria, the size of the genome that also varies, and at least few million base pairs that may be present in a bacterial cell. The nucleid can be clearly visualized by using electron micrograph at high magnification, where it is clearly visible against the cytosol. Sometimes even strands of what is thought to be DNA are visible. The nucleid can also be seen under a light microscope by staining it with the Fulgen stain, which specifically stains the DNA. The DNA intercalating strains, that is DAPI and ethidium bromide, are widely used for fluorescence microscopy of nucleotides. That means when we have to observe the nucleid from bacteria, then there is a use of fluorescent microscopy. That means there is a fluorescence which is generated, and due to which the material, that is the DNA, becomes visible. Experimental evidences suggest that the nucleid is largely composed of about 60% DNA plus a small amount of RNA and protein. That means these are the composition of that nucleid which is present in the bacterial cell. Okay, so the latter two constituents are likely to be mainly messenger RNA. We know that there are three types of RNAs. Which are these three types? That is messenger RNA, transfer RNA and ribosomal RNA mainly present. And here the latter two constituents, that is, are likely to be mainly messenger RNA and the transcription factor proteins found regulating the bacterial genome. Proteins helping to maintain the supercoiled structure of the nucleic acids are known as nucleide proteins. We know that in our bacterial cell, the genetic material, that is DNA, and which is supercoiled structure. Okay, and due to which that supercoiled structure that is maintained by the nucleide proteins or nucleoid associated proteins and are distinct from the histones of eukaryotic nuclei. When we compare the nucleids of bacteria and nucleus of eukaryotes, in eukaryote there is a histone proteins, they are present in the construction of DNA, but in case of prokaryotic DNA there is a nucleide proteins, they are present instead of histone proteins. In contrast to histones, the DNA binding proteins of the nucleide do not form nucleosomes, in which DNA is wrapped around a protein core. Instead, these proteins often use other mechanisms such as DNA looping to promote the compaction. Means long DNA that is fixed in a smaller organism, and that's why there is some looping mechanism or DNA loops they are formed to promote the compaction of the DNA. Now the another material which is present in the cytoplasm or another organelle of the cell, that is the plasmid. Here plasmid is also one type of DNA. Okay, it is also called as a extra chromosomal DNA molecule within a cell that is physically separate from the chromosomal DNA and can replicate independently. Generally, there are two types of DNAs that are present in bacteria, but it is present in some bacteria. The chromosome is present in all bacteria, but plasmid that is present in some bacteria, and that plasmid is having the self-replicating power, or it can replicate independently than the chromosomal DNA. They are most commonly found in small circular double-stranded DNA molecules in bacteria. Okay, in nature, plasmids often carry genes that benefit the survival of the organism and confer the selective advantage such as antibiotic resistance. Plasmids are usually very small and contain only additional genes that may be useful in certain situations or conditions. That means sometimes what happens, these bacteria, they are killed by some substances, and for that purpose there is some genes or there are some plasmids which gives the resistance to that organism from the chemical substance. For example, there is a streptomycin resistant plasmids are there, or antibiotic resistant plasmids, they are present in some bacteria, and due to which what happens, when such a antibiotics comes in contact with these organisms produce some chemicals which will degrade or which will nullify the effect of antibiotics, and that's why these plasmids, they are very, very important to these bacteria. And sometimes these plasmids bear some new characters such as insulin production. If there is a bacteria which has an ability to insulin production and bears the plasmid, and due to presence of plasmid, that organism got an ability to produce insulin, and such a insulin producing organisms, they are useful for the human health, and for that purpose these plasmids, they can be transferred from one cell to another cell for the recombination purpose, and from these microorganisms we can take the insulin. Okay, so with this we finish here today these three organs, that is the Fimbriae and Pili. Then we also study the nuclear material and the plasmid